Welcome to the TOEFL Listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Morales. Thanks for meeting with me during office hours to talk about bioluminescence in ocean depths.
Good afternoon, Mark. I read your abstract and you have interesting questions. Where would you like to start with bioluminescence in the deep sea?
I want to understand both the chemistry that makes organisms glow and the evolutionary reasons they do it in the mesopelagic and bathypelagic zones.
That is a good approach. Chemically, most bioluminescence involves a luciferin substrate and an enzyme called luciferase, or a photoprotein that triggers light emission through an oxidation reaction.
So luciferin is the molecule that reacts, and luciferase speeds up the reaction. Is that correct?
Yes. The enzyme catalyzes oxidation of luciferin, releasing energy as visible light. Some species use photoproteins that bind calcium and undergo a conformational change to emit light, instead of a classic enzyme-substrate mechanism.
Are the luciferins the same across different deep-sea organisms?
No. Interestingly, different taxa use chemically distinct luciferins. There is evidence of convergent evolution because separate lineages evolved bioluminescence independently, using different molecules that produce similar blue-green light.
Why blue-green light specifically?
Because water absorbs longer and shorter wavelengths more strongly. Blue-green wavelengths have the best transmission in seawater, so light at those wavelengths travels farther and is most useful in deep ocean communication or camouflage.
That makes sense. You mentioned convergent evolution. Can you give me a specific example in deep-sea organisms?
Take lanternfish and certain squids, for example. They have photophores distributed along their ventral surfaces to perform counter-illumination. Meanwhile, anglerfish have a photophore at the end of a filament to lure prey. These structures evolved independently in fish and cephalopods.
I have heard about symbiotic bacteria in light organs. Does that occur at depth as well?
Yes. Many deep-sea animals host bioluminescent bacteria in specialized light organs. The bacteria produce light using their own luciferase systems. The host controls light emission by regulating the bacteria population or by using shutters and reflectors.
Do deep-sea bacteria produce the same wavelength as animal-produced light?
Often bacterial light is blue-green too, because that wavelength is advantageous. Hosts may use filters or reflectors to modify the apparent color or directionality to suit camouflage or signaling.
You said counter-illumination earlier. Could you explain how that works in more detail?
Counter-illumination is when an organism matches the light coming from above to erase its silhouette from predators below. Photophores on the ventral side emit light at intensities and wavelengths that reduce contrast against downwelling light.
So the animal adjusts the intensity to match background light. Is this active control?
Yes. Some species sense ambient light and modulate photophore intensity. Others change posture or orientation. Lanternfish perform diel vertical migration and adjust emission as they move through shallower, brighter waters at night and deeper, darker waters during the day.
What about using light to attract mates or warn predators? Are those functions common at depth?
Yes, those are common. Mate attraction often relies on species-specific flash patterns or light displays. Warning or diematic displays are rarer, but some organisms produce sudden bioluminescent clouds or jets to confuse predators.
Could you give me a case study of an ROV observing bioluminescence in situ?
On a recent expedition, we observed a loose-jaw fish emitting directional blue light from suborbital photophores while hunting. The fish adjusted angle and intensity to illuminate prey without revealing itself. Video analysis showed the light matched the spectral window for water transmission and was pulsed in short bursts to reduce energy cost.
Energy cost is an interesting point. How metabolically expensive is producing light?
Bioluminescence is actually efficient in terms of photons produced per unit energy due to high quantum yield of some luciferase reactions. However, there is still a metabolic cost associated with synthesizing luciferins and maintaining photophores or bacterial symbionts.
So the benefits in predation or mating must outweigh the costs in many environments.
Exactly. Natural selection favors bioluminescent traits when the fitness gains from prey capture, evasion, or reproduction exceed energetic costs.
You mentioned directional light and shutters. How do animals control emission physically?
Control mechanisms include muscular shutters that cover photophores, pigmented cells to block or filter light, and reflective tissues that redirect emission. Some species have lens-like organs to focus beams, like the anglerfish lure, which concentrates light to attract curious prey.
Are there genetic studies that show how luciferase genes evolved in deep-sea species?
Yes. Genomic analyses reveal that luciferase and related genes appear in many lineages with structural differences. Comparative studies show convergent amino acid changes in enzymes that tune emission spectra to blue-green wavelengths.
How do researchers measure bioluminescence in situ without disturbing organisms?
Researchers use low-light cameras and red-light illumination that many deep-sea organisms cannot see to minimize disturbance. ROVs sometimes deploy dark chambers to observe natural behavior, and optical sensors record spectra and intensity in real time.
For my project, I was thinking of modeling how depth and water clarity influence optimal emission wavelength. Is that feasible?
Yes. You can model light attenuation with depth using inherent optical properties of water, and then assess how wavelengths attenuate. Combine that with behavioral data on visibility and predator-prey interactions to predict selective pressures.
Would you recommend including examples like anglerfish and lanternfish in the model to validate predictions?
Absolutely. Using case studies like anglerfish lure behavior and lanternfish counter-illumination will ground your model in empirical observations and help test hypotheses about function and evolution.
One last question. Is bioluminescent communication likely to be species-specific in the deep sea given low light conditions?
Yes. Species-specific flash patterns and emission timing are important for mate recognition and territory. In the deep sea, where visual cues are limited, temporal patterns and precise spectral tuning become reliable signals.
Thank you. This helps a lot. I will include chemical mechanisms, spectral transmission, case studies, and evolutionary analysis in my paper.
Good plan. Bring a draft and we will review the modeling assumptions and the way you present evidence from ROV observations and genetic studies.
Listen again to part of the conversation, then answer the question.
Blue-green wavelengths have the best transmission in seawater, so light at those wavelengths travels farther and is most useful in deep ocean communication or camouflage.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit spinta.org.